Good evening, everyone. I will start the lecture in 10 minutes since only few people have joined. I will wait for 10 minutes and then start the lecture at 6:10 p.m. So, are the slides visible? And am I audible? Please confirm. You will. Okay. So, I will now start the presentation. So, welcome all to the week 3 tutorial of Biomass Conversion and Biorefinery. The course coordinator of this subject is Professor Kaustav Mohanty from the Department of Chemical Engineering, IIT Guwahati, and I am your TA for this course, Arihant Ahuja. I'm currently doing PhD in the Department of Paper Technology, IIT Roorkee, and I'm doing PhD under Professor Vibhor Kumar Rastogi, who is in Department of Paper Technology, IIT Roorkee. So before going to the lecture, I would like to share something about me that I've done my BTech in chemical engineering from Comoinjing College, and then did my masters from packaging technology from IIT Roorkee. And currently I'm doing PhD in paper tech, and I'm enrolled in this course since 2020, and I'm receiving the PMRF fellowship during the PhD for the project of paper-based value-added products. So in this today I will discuss about week 3 assignment of 2021, 2022, and 2023. And please try to give the answers. You may know the answer, but you I encourage you to share the answers and don't feel that the answers might be wrong or not. You can make a guess also. So let's start from the assignment 3 of 2021. Who defined the biorefinery as the sustainable processing of biomass into a spectrum of marketable bio-based products and bioenergy. So, which organization has given this definition for biorefinery? Is it NREL, USDOE, IEA Bioenergy Task 42, or CAM et al., a group of people who has done research in 2006? Anyone know the answer? You can share. Yes, the answer is IEA Bioenergy Task 42. So the definitions are more or less same for all these organizations, but they have defined in their own wording. I will share the definition of all these four to you. So IEA Energy Biotask has defined bioenergy as a sustainable processing of biomass into a spectrum of marketable products and energy. It means to make a product marketable by doing a processing in sustainable way. So if we do a processing in sustainable way, and the and such that it comes into the market and it has a value of energy, a good value of energy, then it is known as biorefinery. This definition was given by IEA Bioenergy Task 42. NREL gave the definition of a facility that integrates biomass conversion process and equipment to produce fuel, power, and chemicals. So they said that the bioenergy is nothing but it integrates the biomass with the process and equipment. They just add these two things, that the integration of biomass with the process and equipment to produce some fuel, power, chemicals is known as bioenergy. And the definition given by USDOE is a facility that integrates biomass conversion process and it to produce fuel, power, and bio. So USDOE and NREL both have similar, not similar. They both have same definitions for the biorefinery. And then the group of CAM et al. In 2006 defined bioenergy as sustainable processing of biomass into spectrum, which is similar to that of IEA bioenergy. So the first one to give this definition was IEA bioenergy, and CAM et al. just took their definition and remarketed. So basically, energy is nothing but integration of biomass conversion with the process and equipment in a very sustainable way to make it a marketable product and have energy value. So if I combine these two definitions, we can came up with a new definition also. So these they are just only two definitions, but are shared by four different organizations. So let's move on to the next question. Which biorefinery use wet type of biomass and extract juice from the biomass? Is it green biorefinery, conventional biorefinery, thermomechanical biorefinery, or marine? Can you repeat the answer? It was not audible. So the answer is yes, green biorefinery. So first of all, I will tell you about green biorefinery. Green biorefinery are designed to process wet biomass feedstock such as grass, leaves, and other plant to extract valuable components such as protein, sugar, and fibers. This typically involves mechanical or enzymatic extraction of the juice or sap from the biomass, which can be further processed to make biofuel or biochemist or other any high value compounds. So green biorefinery in simple terms means that we had to take some wet biomass such as grass, leaves, and other plants. So this plant, leaves, and grasses defines the green word here. And biorefinery means to make energy out of it. So the second step is to involve mechanical enzymatic extraction from the to do mechanical or enzymatic extraction from the biomass, which can be further produced biofuels or biochemicals. One example of green biorefinery is the production of ethanol from sugarcane. And in this process, sugarcane is first harvested and then juice is extracted using mechanical presses. So the first step is to mechanically press the sugarcane to produce the juice. And then this sugarcane juice, sorry, and then this sugarcane juice is then fermented to produce ethanol. While the remaining biomass, which is also known as bagasse, can be used for energy production. So this is a typical example of green biorefinery. I will just repeat it again. So green refinery means to produce some type of energy from leaves or grasses or any other kind of plants in its wet form. So how do we take the wet form? We will first take feed. We can do use a mechanical press, and then that mechanical press can be used to extract the juice from the sugarcane, and then this juice will be then fermented to produce ethanol. Now we have utilized the juice of the sugarcane to produce ethanol, which is a fuel, but the remaining mass of the sugarcane, which is also known as bagasse, this can be used again for energy utilization. Here is a typical diagram, a second example. The first example which I showed was sugarcane, and this one involves miscanthus. So miscanthus is a type of grass or crop which we can it is these are very long grasses, and we can use a chipper or a shredder for size reduction. So we can use first step is to reduce the sizes of this grass just by breaking the length using a chipper and a shredder. After that we can press it, and this pressing will result in two things. First is solid cake and second is juice. This juice can be used to make ethanol by fermentation, or we can also use this for algae cultivation, and this algae cultivation can also be used to produce biodiesel. And this solid cake can be again processed further to produce ethanol. So in this way we can produce both bioethanol and biodiesel from some type of plants or crops in its wet and solid form. So this is known as wet biorefinery. So this is known as green biorefinery, and it is used in wet form. Uh, Now, there were three more options for this, which were conventional biorefinery, thermomechanical biorefinery, and marine biorefinery. These are not used in as a wet form, and for example, the conventional biorefinery typically use dry biomass feedstock. These only use dry biomass feedstock such as wood chips or agriculture residue, and employ thermal and chemical conversion to produce biofuel and chemicals. So conventional biorefinery use dry biomass such as woods, and thermomechanical biorefinery use high temperature processes such as gasification or pyrolysis to convert biomass into energy. So thermomechanical is a process in which we use a dry mass and then burn it in the absence of air. So we do the pyrolysis and then convert into energy. And third is marine biorefinery. Marine biorefinery uses seaweeds or other marine biomass as feedstock to produce a very vast range of products such as biofuel, biochemicals, food additives. So let's move on to the third question. Which biomass is not considered for marine biorefinery? Is it microalgae, seaweed, macroalgae, or lignocellulosic biomass? Anyone who wants to guess about it? Anyone? Okay, so there's a hint for it. Marine biorefinery also involves some kind. With if you go for the name marine, you can already guess that it may include seaweeds and algae. So both algae will come. So microalgae, seaweed, and algae, all three are marine biorefinery. And which is not a marine refinery? The remaining, which is lignocellulosic biomass. So, what is lignocellulosic biomass? Lignocellulosic biomass is a complex biomass metal that is composed of lignin, cellulose, and hemicellulose. So, if we go for the word lignocellulose, it is made up of two words, lignin plus cellulose. And this cellulose also includes a part which is known as hemicellulose. So, to define any material which is lignin, cellulose, and hemicellulose, we call it lignocellulose. Lignocellulose are those materials which are mainly plants. These are mainly plant and tree based materials. Lignin, cellulose, and hemicellulose are found in plants, trees, or any kind of leaf also. So, binder, if we go for the options, biorefineries that focus on processing of lignocellulose are known as lignocellulosic biorefinery. So, lignocellulosic biorefineries are the term used for raw material of lignocellulose. And these biorefineries are designed to convert lignocellulosic biomass into a range of value-added products, including biofuel, such as cellulosic ethanol and biodiesel, and some other chemicals also, such as organic acid and ethanol. And a biorefinery that processes lignocellulosic minerals is not considered as conventional biorefinery. Many people get confused in conventional biorefinery and lignocellulosic biorefinery, but these both are not same. A conventional biorefinery utilizes feedstock that are rich in accessible sugar such as corn and sugarcane, and lignocellulosic biorefinery are which involves carbohydrates or such as hemicellulose and cellulose. So many people get confused in conventional biorefinery and lignocellulosic biorefinery. So I hope the confusion you are not in confusion of this. So let's move on to the fourth question. According to IEA T42, following is not considered in four features based new biorefinery classification approach. Is it platform, quality, products, or process? Any guess? If someone wants to make a guess on this, you may speak or write in the textbook. Feel comfortable. Okay, so the answer is quality. Quality is not considered as the four features based on new refinery. The four features that IE consider is platform, products, process, and feedstock. So, according to IEA, the new refinery classification is based on feedstock, platform, process, and products. So, this is the classification approach, and this is the feature which they consider. And quality is not the feature for new refineries, particularly for new refineries. The approach considers the characteristics of feedstock. This approach considers the characters of the feedstock, the platform used to get feedstock, the process employed to refine, and the resulting product. So many people, when they read about this statement, they say that the characteristics of the feedstock is considered. So they guess that it might be quality, and so many people say that all of the above might be the answer. But no, it's not about the quality because when we see the quality, it might be quality of whole, but we are only talking about the feedstock. So the feedstock is the right answer. Okay, let's move on to fifth question. C5 and C6 sugar platform biorefinery for bioethanol from lignocellulosic residue considered as one platform, two platform, three, or multi-platform biorefinery. Anyone? Any guess? So the answer is two platform biorefinery. So first of all, I will tell you what is single platform or two platform. A single platform biorefinery uses single feedstock as its primary input to produce a range of products. So yes, single platform means single feedstock. For example, a corn-based ethanol biorefinery would be considered a single platform because it uses corn. So it has a single input, single feedstock. So it is known as single platform biorefinery. On the other hand, a two platform biorefinery means two different feedstocks to produce range of byproducts. For example, if someone uses corn stover as a feedstock and other as uses switchgrass, just to if they uses two types of feedstocks, as corn stover and switchgrass, then this is known as two platform biorefinery. So in this case, C5 and C6 two types of sugars are used. So it is known as two platform biorefinery. So a C5 platform biorefinery typically focuses on conversion of C5 such as xylose. So they usually work on xylose, and the C6 platform will focus only on the conversion of C6 sugars such as glucose. So both have are different types of sugars. One is xylose and second is glucose. So both will be considered as these two C5 and C6 sugar will be considered as two different feedstock. So this is known as uh, it is not considered as single but as two platform system. So, in this sense, a biorefinery that uses both C5 and C6 sugar in the production of biofuel and the bioproduct could be considered as dual platform biorefinery, which with the separate processing streams for each type of sugar. Now what is C5 and C6 sugar? So C5 and C6 sugar is nothing but a sugar which has 5 carbon in its chemical structure and 6 carbon in its chemical structure. So C6 sugars are more commonly used in the production of biofuels such as bioethanol, as they can be more easily fermented. And but now we have also achieved a good conversion rates for C5 sugar also, to convert it into biofuel as well. So this is a typical type of C5 sugar. If we count the carbon atom here, so it is one, 2, 3, 4, and 5. So this is a typical C5 sugar, and this is a C6 sugar. It has 6 carbon, one, 2, 3, 4, 5, and 6. So this in this way you can identify if a sugar is C5 or C6 sugar. Okay, so let's move on to the sixth question. Syngas is considered as building blocks for what? C1, C2, C3, or C4? Any guess? Okay, first of all, what is syngas? Anyone has any idea? Okay, so actually the answer lies in the syngas definition only. The answer is C1. I will explain you the syngas and C1 and why it is. What are these C1, C2, and C3, C4? What are these? I will explain you in the next slide. So, syngas is typically considered as C1 building block because it composed of primarily single carbon atom. So, syngas, if we see the composition of syngas, it is made up of carbon monoxide and hydrogen. So, it is made up of carbon monoxide and hydrogen. So, this is known as syngas. Now, if we see, it has only one carbon. So, if we make anything out of it, it will have only a single carbon. So it will always be used for C1 as a C1 building block. That's why the answer is C1. So syngas, also known as synthesis gas, is a fuel mixture consisting of primarily hydrogen and carbon monoxide, and it is produced by gasification of biomass, coal, or other carbon-based feedstocks. And this is the typical structure of syngas: CO plus H2, carbon monoxide, and hydrogen. Now, if we go for what is C1, C2, C3, and C4, and what type of compounds are they do they require? So C1 refers to a single carbon atom or molecule such as methane. So C1 means single carbon atom, and one example is methane. Other is carbon monoxide, carbon dioxide, and methanol. So if we talk about any gas, any type of gas which has only one carbon in it, so it will be said as a C1 building block. In case of C2, it will have two carbons, so such as ethylene, ethane, and acetylene. Both have two carbons, which is this is uh, this represents C2 building block. And similar cases are with C3 and C4. C3 refers to building blocks containing three carbon atoms such as polypropylene and propane, and C4 refers to building blocks containing four carbon atoms such as butadiene and butene. So, by looking at this C1, C2, and C3, C4, now everyone is clear that syngas is made up of CO plus H2, so it has only one carbon, and thus it will be referred as C1 building block. Okay, so let's move on to the next question. Which content is most abundant in plant biomass? Hydrogen, carbon, nitrogen, oxygen. Anyone wants to make any guess about it? You may know the answer also, but if you don't know any type of guess, do you want to make it? Anyone? Okay, so no guesses. Hmm. The answer is carbon. Most of the plant-based compounds are rich in carbon and oxygen, but carbon is at highest concentration. So carbon is the most abundant element in the plants. It is a key component to organic molecules such as carbohydrate, lipid, protein, and nucleic acids. So every kind of compounds present in plants such as carbohydrate, lipid, proteins, nucleic acid are made up of carbons, such as every carbohydrate, every kind of sugar, starch. All these compounds have carbon inside it. When we look for the hydrogen and oxygen, that in what form they are present, so they are essentially present as a component of water. Majorly they are present in form of water, H2O, and oxygen is present in form of oxygen and hydrogen are also present in form of hydroxyl bonds, OH bonds. As we have seen that all these carbohydrates has a OH bond in it. So this OH bond is also a source of hydrogen and oxygen in plants. And third, nitrogen. Nitrogen is also important for plant growth. It is a key component for amino acid and nucleic acid, but it is very less abundant than carbon, hydrogen, and oxygen. Nitrogen is very limited. It only makes up essential amino acids and nucleic acid required for the plants. And at last, sulfur. Sulfur is also present in plant tissues but in very smaller quantities and is also important for synthesis of certain amino acids and proteins in the same way as nitrogen. So the major component in plants is carbon and then hydrogen and oxygen, and further nitrogen and sulfur is the least present in the plant. Okay, so let's move on to the eighth question. Which is not considered as macronutrient for plants? Potassium, nitrogen, boron, or phosphorus. So the term here is macronutrients. There are two types of nutrients which plants require. One is macro and micro. Other is micro. So which is not considered as macro? Any guess? Arthi, Vijay, any type of guess you want to make? Boron, correct. So boron is not considered as macronutrient. It is the right answer. And before going to it, I will tell you about something about the NPKs. You might have also heard about the term NPK. NPK is a very vast term used in fertilizers, which represents nitrogen, phosphorus, and potassium. So these three NPK are considered as macronutrients, which were the first three options. So yes, NPK are the macronutrients which are required in very large amount by the plants. So many fertilizers are made up of NPK. And some other nutrients are also known as macronutrient, and they are such as calcium, magnesium, and sulfur. These also required by some plants. But other than these macronutrient, there are some micronutrient. Micro, as the word comes, micro means small. So these are not required in large amount. These are required in low amount. So these are known as micronutrients. So micronutrients are required in smaller quantities by plants, and these includes iron, magnesium, zinc, copper, boron, molybdenum, or chlorine. So boron is a micronutrient, not a macro. So yes, Vijay was right. Boron is the correct answer. And here is a list of essential plants, essential micronutrients and macronutrients required by plants. So we have seen that NPK are the macronutrient and CHO, which make up most of the plant, carbon, hydrogen, and oxygen, as we have seen in the last questions also. These make up a major part of plants. So these are also required required for the plants. And other macro elements are calcium, magnesium, and sulfur. And these are the rest micronutrients which are required for the plants. And we can also define these macronutrient and micronutrient based on mineral waste and non-mineral waste. So the CHO, carbon, hydrogen, and oxygen, which we have seen, which these are the major composition of the plants. These are also known as non-mineral based nutrients. Does not have any minerals in it. These are the major building blocks of the plant, and the rest of the elements which are discussed are mineral based compounds. These provide some minerals to the plant, and every type of other macronutrients or micronutrients are mineral based. So let's move on to the ninth question. Sucrose sugar is considered as monosaccharide, disaccharide, trisaccharide, or polysaccharide. Any guess? Okay, monosaccharide. Anyone wants to make? Anyone else? Okay, so actually the answer is disaccharide. Correct answer is disaccharide. Monosaccharide is not a right answer. I will explain this in the next slide. So first of all, yes, yes. Okay. And in NPTL assignment 3. Okay. Yes. I know that there is some mistake about at that point. The correct answer is disaccharide, and yes, it is showing monosaccharide as the answer. We will contact the NPTL team, and then we will request them to make it correct. Okay. So, for to go into deeper meaning about the monosaccharide and disaccharide, I will explain this in very simple terms. So monosaccharide means there is a term in this which is mono. So mono means single compound, and saccharide means sugar. So monosaccharide means a sugar compound which has a single molecule. So some examples of monosaccharides are glucose, fructose, and galactose. And disaccharides means di. The word here di means two. So disaccharide are made up of two monosaccharides compound, and these are joined. These two monosaccharide bonds, these two monosaccharide rings are joined by some type of bond, and this bond is known as glycosidic bond. So some example of disaccharide is sucrose, lactose, and maltose. So yes, sucrose is the right answer because sucrose is made up of glucose and fructose. And if we go to trisaccharide again, we if we go for the words, tri means three, so it is made up of three monosaccharides, and all these three monosaccharides are bonded with glycosidic bonds. And the example of trisaccharide is raffinose, which is also composed of galactose, glucose, and fructose. So you don't need to go into the example, just go into the concept of it, that what does is it mean? Mono, di, and tri. And at last, another example is polysaccharide. Polysaccharides are made of many types of monosaccharides joined together. So poly means many. So this is the example of monosaccharide. So it is a type of monosaccharide, and this is also a type of monosaccharide. But if we join these two, this bond, COC linkage, this COC linkage, also known as glycosidic bond, joins these two monosaccharide, and thus we get disaccharide. So this is what glycosidic bonds look like. So this is known as carbon. It is C, and it is again COC. So it is a COC bond, also called as ether linkage and also known as glycosidic linkage. So sucrose is made up of two types of monosaccharide. That's why it is called as disaccharide. And who have selected disaccharide and the answer is showing monosaccharide need not worry. We will contact the NPTEL team and request them to correct the answers. Okay, let's move on to the 10th question of 2021, which requires strong acid or bases or any other type of hydrothermal treatment to be hydrolyzed. Is it protein, lipid, cellulose, or lignin? Which of these require a strong acid or base or very harsh hydrothermal treatment to get hydrolyzed? Any guess? Yes, the answer given by NPTL is lignin in this case, but actual answer should be cellulose in this case also. And I will tell you why about this. So first of all, cellulose is a very highly crystalline molecule that is tightly packed and protected by hydrogen bonds between its glucose monomers. It is a very crystalline molecule. So, crystalline means that every atom in it is very tightly packed and it is in very ordered manner. So, everything, if it is in very ordered manner, any type of acid or alkali attack is not possible at mild condition. So we have to uh, do a very good. We have to use very harsh acid and alkali to hydrolyze it. And there is lignin also present in above inside it. So lignin protects these cellulose. So yes, if the answer of the question might be lignin if we twist the language of it, that the presence of lignin will inhibit the hydrolysis of cellulose, but the hydrolysis of cellulose is very difficult. So the question is very twisted in its language, uh, but basically I want to tell you is that in order to make any biofuel, we have to hydrolyze cellulose, and cellulose does not hydrolyze easily. We need very high amount of H2O, SO4, or alkaline treatment to hydrolyze it. And lignin is also present, which is blocking it. So we have to remove the lignin also from the cellulose in order to hydrolyze the cellulose. So, as a result of high crystallinity of cellulose, it is highly resistant to degradation and requires strong acid and bases or harsh hydrothermal treatment to be hydrolyzed to convert it into glucose monomers. Lignin, on the other hand, is typically removed from bioprocess known as delignification. So there is a process known as delignification, which can be done with the help of soda pulping and some type of organosol pulping also. Lignin can be removed very easily by chemical, mechanical, or enzymatic treatments. So, to make a biofuel from the plants, we have to first remove the lignin, then do very harsh hydrolysis on the cellulose. And on the other hand, protein can be hydrolyzed using enzyme or acid hydrolysis, and lipids can be hydrolyzed using enzymes or through transesterification. So, proteins, lipids, all and lignins all can be hydrolyzed easily, but cellulose requires very high amount of acids and very high temperatures to degrade or to make glucose monomers out of it. So now let's move on to the assignment 3 of the year 2022. We have discussed the assignments of 2021 which were similar to that of 2023 also this time, and now we will discuss about 2022. So the first question, which technology do not come under biochemical based treatment process, which is not a biochemical process in this, any type of graph? Yes, correct answer. Pyrolysis is not a biochemical process. Basically, pyrolysis is a method to produce um, high quality fuel. In this process, biomass is stored in a very um, biomass is stored in a chamber in which vacuum is created, and it is heated in the presence of vacuum. All the in Not in the presence of air. Air is totally removed, and during this process we get some type of fuel in the form of vapor or gas, and below it some kind of char is also uh, produced. So this vapor or gas is taken out from this tank, and it can be used as a fuel. So this is known as pyrolysis, and it is a type of thermo uh, thermochemical. No, it is a type of thermal method to produce. It is not a biochemical method. So, pyrolysis is a thermochemical process that involves heating biomass in the absence of oxygen. The major thing about the pyrolysis is that we heat in the absence of oxygen to produce mixture of solid char, liquid biooil, and combustible gases. This process is distinct from biochemical process like fermentation, anaerobic digestion, which involves use of microorganisms to convert biomass into fuel. So fermentation and anaerobic digestion involves the usage of some type of enzymes or microorganisms. These enzymes and microorganisms feed onto the biomass, and then they produce some kind of useful products which can be converted into energy. So the use of microorganisms and enzymes, this defines this process as biochemical process. So fermentation and anaerobic are biochemical, but pyrolysis is not biochemical. And if we go into more details, fermentation is a process in which microorganism like yeast and bacteria is used to break down carbohydrate into similar compounds such as alcohol and organic. And anaerobic digestion is also a similar process. It also takes place in the absence of oxygen, but this also requires some type of microorganisms. So microorganisms will be used for anaerobic digestion in the absence of oxygen, but due to the presence of microorganisms it is known as biochemical. So anaerobic digestion, aerobic digestion, fermentation, all three are biochemical, and pyrolysis is a thermochemical because it involves heat and absence of oxygen. So now let's move on to the second question of 2022. Hemicellulose polysaccharide is made up of which sugar? Pentose, hexose, both, or none of the above? Any guess? Okay, no guesses. So the answer is both. Hemicellulose is made up of pentose and hexose both. And if we go on the structure of hemicellulose, it is a type of it contains two different things. One is pentose and second is hexose. So what are these pentose and hexose? Pentose is nothing but carbohydrate with five carbons, as the name goes, pent. So one, two, three, four, and five. It has five carbon atoms, so the name is pentose, and it also contains hexose. Hexose means six, so it has six. One, two, three, four, five, and six. It has six carbon atoms, so it is termed as hexose. Hemicellulose is made up of both of these compounds, pentose and hexose, and joined by some kind of linkages, and it is a repeating unit order. So it is a type of polysaccharide because this N means many types. So pentose and hexose are both types of sugar that are found in different things. Pentose has 5 parts in it, as you have seen in this diagram also, while hexose has 6 parts in it. They are both building blocks, and DNA, which we know that DNA is produced, is made of pentose, and fruits or honey is made up of hexose. So these pentose and hexose are the building blocks of bigger compounds such as DNA or fruits or honey. So till now we have seen that hemicellulose is a polysaccharide that is composed of different mixture of sugars including pentose and hexose, and there are many types of pentose also, such as xylose and arabinose, and many types of hexanose such as glucose and mannose. So it is a mixture of pentose and hexose, and it has many repeating unit. So it is known as polysaccharide. The precise composition of hemicellulose depends vary on the source of biomass, but it contains higher proportion of pentose than hexose. So particularly, we know that pentose is higher than hexose, but by what amount and how much it is higher, this always vary with every kind of source. Different types of plants, different types of stem, leaf, bark, root, or any kind of shrub, tree will result in different ratio of pentose to hexose. So we cannot define the ratios, but generally we have seen that pentose content is higher than the hexose in case of hemicellulose. So this makes hemicellulose an important potential source for pentose sugar for the production of biofuels and other value added products. So we know that it has pentose sugar in it. So we can utilize this pentose to produce some kind of biofuels or any kind of value added products. So that's why there is a lot of research going on the hemicellulose utilization. So now let's move on to the third question. Which of the following options arrange correctly regarding elemental composition of plant biomass? So, among these, which is the correct order of hydrogen, oxygen, sulfur, and carbon content inside a plant? Can you repeat? It was not audible. Okay. So, yes, carbon, oxygen, hydrogen, sulfur is the correct answer. So, carbon is the most abundant element in plants, followed by oxygen and hydrogen. And nitrogen is also present. In this case, nitrogen was not written, but I'm just telling that about nitrogen because nitrogen exists here between hydrogen and sulfur. So if we go into more deeper composition, carbon is the highest, which is the major building block. Everything is made up of carbon, and oxygen and hydrogen is present in uh, the hydroxyl groups and in the presence of water inside the plants, and at the least is sulfur. But before sulfur we have also nitrogen. So nitrogen is also present in plants in significant quantities but smaller than carbon, oxygen and hydrogen. And sulfur is present in very smaller quantities. So yes, you are right, the correct answer is carbon, oxygen, hydrogen, sulfur. So, what are the uses of these, or what role do these compounds play? So, if we look at the carbon, we know that everything is made up of carbon, so it is the building block of all organic molecule in plants, including carbohydrate, protein, nucleic acid, and lipids. So, any kind of thing which we see or in the plants, if we see the composition, it is either carbohydrate, protein, nucleic acid, or lipids. Everything has carbon inside it. So, through photosynthesis, plants convert carbon dioxide from atmosphere into organic compound and they use it for energy and growth. So, they utilize the CO2 from the environment and convert this into the energy, and utilize this energy for its own growth. So carbon is the major building block of plants. If we go for the oxygen, oxygen is essential for respiration of plants, where it is used to break down organic molecule to release energy. And the major in if we look for the major structure of any type of sugar such as pentose, hexose, or any type of glucose sugar, everything has OH in it. So this OH, also known as hydroxyl bond, this is made up of oxygen and hydrogen. So the third thing, hydrogen is a key component of water also. So plant also has water in it, and it uses water for its own growth. It takes up water from the roots and provides it everywhere. So hydrogen is key component of water, which is essential for plant survival and growth, and it is a component of organic molecule in plants, including carbohydrate, proteins, and lipids. So now let's move on to the fourth question. Which carbohydrate comes under polysaccharide category? Which is a polysaccharide? Is cellulose, hemicellulose, starch, or all? Yes, correct answer. All of the above are polysaccharide. So by now you know that polysaccharide is nothing but glucose, or you can say five or six carbon member ring with different linkages, with more than three or many linkages. It is a polysaccharide. So cellulose, hemicellulose, and starch. If you look at these structure, these all are polysaccharides. So polysaccharide are complex carbohydrate made up of multiple monosaccharide. And we have also discussed this in previous answers also. And these are linked together through glycosidic bonds. These glycosidic bonds are also known as ether bonds, which represent COC. They are usually large molecules with high molecular weight and can have variety of structural and functional roles in living organisms. Polysaccharides are commonly found in plants, animal, microorganisms, and serve various functions such as energy storage, structural strength, cell signaling, immune system, and response. And major examples of polysaccharide in every type of organism is starch, glycogen, cellulose, chitin, agarose. Polysaccharide also play important role in various applications in such as food, pharma, biotechnology. We also utilize this type of polysaccharide for ourselves also in food, pharma, and biotechnology. And these are also required by the plants. For example, cellulose. Cellulose is a type of polysaccharide, and it can be converted into biofuel and other valuable products through processing through different process of biorefinery. So typically, a typical structure of polysaccharide looks like this. It has a repeating unit of N because it has, that's why the name poly has come in, and saccharides is a repeating unit inside it. This is a typical cellulose and hemicellulose structure, and this is a starch structure. So these all are polysaccharides. These all have repeating unit N in it. So these are polysaccharides. These are not disaccharide or monosaccharides. These all are polysaccharides. Now let's move on to the next question. Which component of lignocellulosic biomass hinder its biorefinery process mainly? We have also discussed similar type of question in 2021 assignment also, so you might answer it. So yes, lignin. So lignin is the compound of biomass that often hinders biorefinery process. Lignin is a very complex polymer that provides structural support to plant cell and is present in high concentration in many type of biomasses. So any type of plant, if we will see the chemical composition of it, it will have cellulose, hemicellulose, and lignin. So lignin is present in very variable quantity of different plants. It may be less than 10% in case of hemp and in hemp plant, but it may be greater than 20 or 40% in different other plants. And every time lignin has a very different chemical structure, so we cannot define a chemical structure to lignin, but it is a very vital chemical for plant because it provides a good structural support to the plant cell. And it also protects the cellulose present in the plant cell for so this has to be removed in order to convert cellulose in any type of form. So this its presence can interfere with the extraction and conversion of other valuable components such as cellulose and hemicellulose. And lignin is also difficult to break down and requires harsh chemical treatment, which can be expensive and environmental friendly. But now pulping is used. We can also do this by mechanical pulping, or we can remove the lignin completely. And also we have different types of methods such as soda pulping, alkali pulping, and organosol pulping. So by doing any kind of pulping, we can reduce the amount of lignin from the plant. So this is a typical structure if we look into the wood. If we look at the microfibers of the wood, it contains hemicellulose, cellulose, and lignin, all. So these three are intertwined in itself. And to if we want to produce fuel out of the cellulose and hemicellulose, if we want to convert, we just need hemicellulose and cellulose to produce fuel. But this lignin is intertwining inside the cellulose and hemicellulose, and it is protecting them. So we need to remove this lignin first so that we can utilize hemicellulose and cellulose to produce biofuels. So the sixth question is for bioethanol. For bioethanol production, which is the main constituent of biomass? Is it starch, lipid, protein, or lignin? Which of these is used for bioethanol production? Any guess? Starch. Correct answer. Starch is used as is the main constituent of biomass, which can be used to produce bioethanol. So, if the main constituent of bioethanol production is starch, which is a carbohydrate, and it is very much abundant in corn, wheat, and barley. So, if we at the look at the bioethanol can be only made by carbohydrate. And among all the options given, only starch was a carbohydrate. So starch is the right answer. This starch can be hydrolyzed into glucose. So we can hydrolyze it and form glucose. And this glucose is a monomer. So we have broken down the starch, which was a polymer, and now we have broken the polymer into the monomer using hydrolyzation method. And this monomer can now be fermented by yeast to produce bioethanol. So it is a two-step method. First is hydrolyzation and second is fermentation. So fermentation is to hydrolyzation is to make starch into glucose, and then this glucose can be fermented by yeast to produce ethanol. So apart from the starch, cellulose, hemicellulose are also very much abundant in lignocellulosic biomass. So hemicellulose and cellulose can also be used to produce bioethanol with the similar but more complex biochemical processes. So we can produce bioethanol from starch also, and from cellulose and hemicellulose also. Just because all these three are polysaccharides, and they are made up of carbohydrates. Now let's move on to the seventh question. Which fatty acid chain ester is most suitable for biodiesel production? So first of all, I will tell you about what is fatty acid chain. So fatty acid is nothing but a very long chain of carbon. Fatty means a very long chain of carbon, and acid means there is a carboxylic acid at the end, COOH. So fatty acids mean a very long chain of carbon. Fatty means a long chain of carbon, and fatty acid means a long chain of carbon with carboxylic acid. So COOH is known as carboxylic acid, and fatty acid chain ester. The ester word here means that this acid COH might be changed into COOR. So, among these C combination, which of the combination is most suitable for biodiesel production? Anyone wants to make any type of guess? Anyone? Last one. Okay. So, the answer for this is C69C18. I will tell you why. So, the most suitable fatty acid chain for biodiesel production is straight chain alkyl ester with 16 or 18 carbon atoms, which is also known as fatty acid methyl ester, also, and in short we call it FAME. Longer, and if we with the longer chain fatty acids tend to have higher melting point, which causes problem in cold weather, while shorter chain fatty acids have lower energy density and lead to reduced fuel efficiency. So if we go for C12 or C14, these are known as short chain fatty acids. So these short chain fatty ester will produce very low energy density, and it will have a very reduced fuel efficiency. So we cannot go for C12 or C40, and we also cannot go for C20 or C24 or C22, any very higher chain, because higher chain fatty acid will be solid at room temperature. So we need something which is in between these two, which is liquid at room temperature and is not very short. So we go generally use C16 or C18 carbon atoms chain. And there is also one more difference in this, that is saturated. There are two types of fatty acid. One is saturated and other is unsaturated. So we usually prefer saturated fatty acids over unsaturated fatty acid for biodiesel production, as these are less prone to oxidation. I will tell you this saturated and unsaturated in the next slide. So first of all, what is C16 and C18? C16 is nothing but it has 16 carbons in it, and C18 means it has 18 carbons in it. And zero means C16 zero. C16 zero means it does not have any C double bond C. It does not have any. And C18 zero also means that it does not have any double bond carbon. It has zero to C double bond C. So there are two types of chains. One is saturated and other is unsaturated. Saturated means there are no C. There is no type of C double bond C. And unsaturated means it has a bond which has C double bond C. So we now know that why we have to use C16 and C18. C16 and C18 is um, uh, it is in between lower fatty acid and higher fatty acid, so it provides a balance of melting point and balance of energy value. But we do not use C, we will not prefer C18-1 or C18-2. We'll prefer C18-0. Why? Because if we it has C double bond C in it. C double bond C means C18 1. 1 means there is one single bond. So in the presence of oxygen, if we will keep this in at room or any open environment, the oxygen from environment will come and it will oxidize this bond easily, and this bond will break down very easily. So the purpose of this fuel will get very low. So that's why the preference of unsaturated fatty ester is less, and we will only use saturated fatty acid. So that's why we majorly use C16 and C18. I hope that you all have understood that why C16 C18 are used, why not C12, why not C24, or why not C16 C18. I hope this concept has been clear now. Any questions in this? If you want, I can repeat this also because it is more about chemistry. If you want to repeat, I can repeat. Okay, I hope that everyone has understood it. So I will move on to the next slide. So this is a typical composition of biodiesel, which varies from feedstock. So it has majorly palmitic and stearic acid, which are C16 and C18. So C16 is known as palmitic acid and C18 is known as stearic acid. And it also might have some unsaturated compounds also, such as oleic acid, linoleic acid, or linoleic acid. C18 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 means these are unsaturated. C18 means one there is a one double bond in it. Two means it has carbon double bond in it, and three means it has three. So these will be in very low amount. Then we can say that it is a good fuel. But if these are in high amount, then these two, then it might not be a good fuel. So palmitic acid and stearic acid are saturated fatty acid, while oleic, linoleic, and linoleic acid are unsaturated fatty acid. So the specific proportion of fatty acid in biodiesel can affect its physical chemical properties such as viscosity and oxidative stability. So to make a better biodiesel of it, we need much higher these two compounds and less of these three compounds in it. So now let's move on to the eighth question. Which of the following are correct stepwise process in processing of lignocellulosic biomass in biorefinery approach? So, which is the best approach uh, to convert into fuel? So, it is pre-treatment followed by separation and hydrolysis followed by pre-treatment, or separation followed by hydrolysis. You can just make a guess if you don't know. Anyone wants to make any type of guess? Correct. So pre-treatment followed by separation and followed by hydrolysis is the right answer. So first we need to pre-treat this lignocellulosic biomass to remove some lignin out of it, and then we can separate it using some type of mechanical or washing, and then finally we can hydrolyze the cellulose and hemicellulose to produce fuel. So pre-treatment involves some kind of chemical treatment to the lignocellulosic biomass. So we have to treat with some chemicals. So chemical treatment or pre-treatment is the first step, and it is done to remove the lignin out of it. So the first step is pre-treatment, which involves physical, chemical, or biological treatment, and the aim of pre-treatment is to break down the complex lignocellulose structure and increase the accessibility of enzymes to the cellulose and hemicellulose fraction. So, our main motive is to make enzyme accessible to cellulose and hemicellulose, and to make it more accessible, we need to remove the lignin out of it. So the main aim is just to remove the lignin from the lignocellulosic biomass and other kind of extractives. There are many more types of lipids inside it, so we need to remove those compounds. So that is known as pre-treatment, and this pre-treatment depends on the biomass feedstock and desired product. So different kinds of lignocellulosic biomass had different kinds of lignin. If biomass have very high lignin, we will do chemical pretreatment, and this chemical pretreatment can be done with pulping. Pulping is nothing but we cook the biomass in the presence. We cook the biomass in presence of heat and pressure with some chemicals in it. So this chemical is majorly soda, NaOH, and that's why it's known as soda pulping. And there is another method also in which we use acetic acid. We can also use acetic acid and then heat the biomass in the acetic acid. And in this way, acetic acid dissolves the lignin. Acetic acid will dissolve all the lignin in the presence of heat and pressure, and lignin will come out and in the acetic acid, and the biomass will be free from the lignin. So these two will can be done if lignin is very much higher. But if lignin is in less amount in the biomass, we can use simple mechanical techniques also. Mechanical technique or just water treatment. We can also use uh, hydrothermal treatment. Hydrothermal treatment means to cook the biomass in the presence of high pressure and temperature, but in the presence of only water. So this can be done if the lignin is less than 5 or 10%. And the second step is separation. This separation means that now we have to remove all the chemicals and lignin f- from the hemicellulose and cellulose. So this can be done by simple filtration or washing also. So after pre-treatment, the biomass is separated into different components such as cellulose, hemicellulose, lignin, and extractive. The lignocellulosic is now uh, separated into all its components such as cellulose, hemicellulose, lignin, and extractives. And we will take this cellulose and hemicellulose further to convert it into fuel. So this separation is easy and can be achieved by mechanical or chemical methods depend upon the biomass and the desired products. And finally, we do the hydrolysis. Hydrolysis is the last step in this after the pre-treatment and separation. Now we have cellulose with us, cellulose or hemicellulose. We can now easily do the hydrolysis of cellulose because there is no lignin, and lignin will not hinder the uh, process of hydrolysis. So the next step is hydrolysis. It involves breakdown of cellulose and hemicellulose into their constituent sugars. So hy- hydrolysis does nothing but it break down the cellulose into sugar. So we know that cellulose is a polysaccharide. So we just break down this polysaccharide into mono or disaccharide, whatever it has. If it is made of monosaccharide, we will break down the polysaccharide into monosaccharide. If something is made up of disaccharide, we will break down in disaccharide. So majorly we break down the cellulose and hemicellulose into their constituent sugars, mostly disaccharides. And this sugars can be easily then further fermented in the presence of enzymes or other microorganisms to produce bioethanol. This process is typically achieved by enzymatic hydrolysis, alkali hydrolysis, or acid hydrolysis, where cellulose and hemicellulose are converted into glucose and other sugars. So this hydrolysis can be done by enzymes, alkali, or acid hydrolysis, but this require a very large amount of acid and alkali hydrolysis. So right now there is a lot of research going on to the enzymatic hydrolysis, so we can reduce the consumption of these high alkali and acid concentration, and also reduce the cost and power consumption because enzymes make it easy. So there are many hybrid methods also which involves both combination of these methods. And this sugar, the obtained sugar after hydrolysis, can be further processed to produce a range of valuable products such as biofuel, chemicals, and all other value-added materials. So now let's move on to the ninth question. for high protein and carbohydrate rich biomass, which is the most suitable biorefinery approach? So for now we have seen that if any if there is a material which is rich in carbohydrate, we can produce a fuel out of it. But if a carbohydrate has protein in it also, also has high protein, then which is will be the most used? Which separation technique will be used? Will we separate the protein and then produce ethanol, or we will first produce ethanol and then separate protein, or the similar ways just to produce biodiesel? Any guess that in case of presence of protein, what will we do? Any guess? Yes, correct. First is correct answer. We will first separate protein and then produce bioethanol. So this is because proteins are very easily separated from carbohydrates. First of all, we can all separate the proteins very easily from the carbohydrates, and so we will first remove the proteins. And after protein separation, the remaining biomass can be used for bioethanol production through hydrolysis or fermentation, which we have also discussed. And but after we will not separate protein after bioethanol, because if during the bioethanol production uh, many type of thermal treatment will be done, and this thermal treatment on the protein will make it denatured, and this will hinder the performance of um, the conversion technique. We will also get less amount of fuel if protein is present, and also the protein will get denatured, so we will not get good quality of protein also. That's why we will first separate the protein, and then we will do the hydrolysis and get the biofuels. Now, now we have seen that protein separation is must first. It will be done first, but then we will do bioethanol. We'll produce bioethanol from it, not biodiesel. Biodiesel we cannot produce from carbohydrates, because biodiesel is produced from lipids. As we have seen that biodiesel need C16 and C18. C16, C18 fatty acid are lipids, and carbohydrate does not produce uh, diesel. It will carbohydrate will produce ethanol only. So we will first remove the protein out of it, and then we will use the carbohydrate to produce ethanol. So the correct answer was to remove the protein and then go for ethanol production. So yes, as we have discussed, protein presence will inhibit ethanol production and may lead to lower ethanol yield, as a protein fraction can inhibit the fermentation process. And biodiesel process is not feasible for carbohydrates, because biodiesel is mainly produced from lipids and not from protein and carbohydrate. So we will only use this if biomass is rich in protein and carbohydrate. We will remove the protein and produce ethanol out of the carbohydrate. Okay, so now let's move on to the last question. Which biomass is not suitable for two platform sugar based biorefinery? So, is it carbohydrate rich biomass, high carbon and hydrogen rich biomass, oil rich biomass, or nitrogen and sulfur rich biomass? So, I will repeat the question: which biomass is not suitable? The question is not suitable. Any guess? Correct. Nitrogen and sulfur is the right answer. So, nitrogen and sulfur rich biomass is not suitable for two platform sugar-based biorefining, as they contain high amount of heteroatoms and can cause issue during the fermentation process. So, if a biomass has nitrogen in it or sulfur in it, during the fermentation process, these will inhibit the fermentation, and so these cannot be used for two platform sugar-based refinery. And a two-platform sugar-based refinery is a process that involves breakdown of biomass such as agriculture waste into individual components, mainly carbohydrate and lignin. And two-platform, just for the information only, that so that you can understand that two-platform is a biorefinery is a platform, means that there are two different types of feedstock. There are two different types of feedstock, so that's why it's known as two-platform sugar-based refinery. So some types of biomasses are not suitable for this process. For example, biomass that is high in oil, nitrogen, and sulfur. See, the if a compound is rich in higher in nitrogen, sulfur, and as well as oil, they might not be suitable for this type of process, as they can reduce the efficiency. So, and majorly this is because the chemical composition of these types of biomass is different and may require different methods to breaking down. So, nitrogen and sulfur-based compounds or oil-based compounds, they will interfere the uh, chemical breakdown of the carbohydrates into the biofuels, and these are the breaking of nitrogen and sulfur are also different. We need different other methods to break down the oil, nitrogen, sulfur, so we cannot go for this method. So, in simpler terms, if we go, not all types of waste can be used in the same way to make biofuel. So, we cannot say that this is also a waste. If there is a waste A and we use hydrolysis for it, and there is a waste B, we will also use hydrolysis for it. So, there is uh, no surety that same method will work for B also. So different types of waste will cannot be used for same to produce same biofuels or with similar matter. Some types of waste are better suited for different methods of conversion, and scientists needs to consider this composition of the waste before choosing the best method. So the waste analysis is also important before converting it into the fuel. So now I will move on to the assignment 3 questions. So most of the assignment 3 questions were related to assignment 1 question. Only there 2 3 questions were different. So I will discuss that 2 3 questions only, because we have seen discussed that in assignment 1 discussion also. And who have joined late this meeting and might not have seen the discussion of assignment 1, assignment 3 of 2021, they can see this recording in YouTube, and the YouTube link will be shared by the NPTEL team. So the fourth question of 2023 this year was, which of the following describe the treatment of biomass by chemical process? So which of these is a chemical process? Hydrolysis, transesterification, hydrogenation, or all of the above? You might have done these questions. So, and yes, yes, all of the above. So let's move on to that, why all are chemical processes. So hydrogenation is a chemical process that involves the reaction of hydrogen gas with the substance in the presence of catalyst, usually high temperature. So if we do the hydrogenation of a biomass, we need hydrogen gas for this chemical process. So the utilization of chemical is there. So that's why it's called chemical process. And in biomass conversion, particularly, hydrogen can be used to convert lignocellulosic biomass into liquid fuels and chemicals. And during hydrogenation, the biomass is typically subject to high pressure and high temperature. So in hydrogenation, what we do is we give very high temperature to the biomass and at a very high pressure. This high pressure and temperature in the presence of hydrogen. So this high pressure and temperature in the presence of a hydrogen and some catalyst, there is some catalyst used. This breaks down the complex biomass molecule into simpler molecules such as alkanes, alcohol, and organic acid. So this is just some basic of hydrogenation in layman terms, that what is hydrogenation and why it is known as chemical process. And when we consider hydrolysis and transesterification, these are also chemical process, but they can also be categorized as biochemical process because they involve the use of biological catalysts such as enzyme to catalyze the reaction. So biochemical hydrolysis and transesterification, these two are also chemical processes, but they can also be biochemical process. So why do we when do we will call it biochemical process? We will call it biochemical process if the catalyst uses of biological terms, so such as enzymes. So if we use enzyme as a catalyst, we will call these reactions as biochemical. So in hydrolysis, particularly hydrolysis, enzymes we use if we use enzymes, it will break down the complex molecules such as carbohydrate into simple sugar. While in transesterification, enzyme catalyze the conversion of triglycerides into biodiesel. So both are different reactions. In one, both involves the usage of enzymes. Hydrolysis also use enzyme. Transesterification also use enzyme. But one converts carbohydrate into smaller sugar, and one converts to biodiesel. So hydrolysis and transesterification both are chemical processes and can also be considered as biochemical process if we use the enzyme instead of catalyst. So these processes are considered biochemical because they involve the use of enzymes which are produced by living organisms to facilitate the reactions. So the sixth question, which of the following is not part of a thermomechanical processes? So out of these four, which is not a thermochemical process? So if we look at the word thermochemical, it means heat plus chemical. So which is not a thermochemical process? Hydrothermal supercritical pulping. Yes, pulping is the correct answer. Pulping is not a part of thermochemical process. It uses chemicals to break down wood and to make paper and products, but it also utilizes some kind of pressure to convert biomasses, and it also may require only mechanical process. We can also do only mechanical pulping to produce cellulosic biomass out of it. So mechanical pulping does not require any kind of heat or chemical if you use to produce uh, cellulosic biomass from it. Whereas hydrothermal, supercritical, and gasification, these all are thermochemical processes. If we go into the basics of these three, hydrothermal process, as the name goes, hydro and thermal, it utilizes heat to water and heat to do the reaction. So it is a method of breaking down organic materials such as biomass in the presence of high temperature, high pressure water. So thermal means high temperature and hydro means water, but in the presence of high pressure. So in the presence of high temperature and high pressure water, the biomasses can be broken down into organic material. So every process, this process also, hydrothermal process is also used to break down the biomass. It is used to convert complex organic molecule in biomass into simple molecules. And supercritical process involves substance is heated and pressurized to a point where it becomes supercritical fluid, which has properties of both liquid and gas. So we pressurize a material and heat it simultaneously so that forms a supercritical fluid. Now, what is supercritical fluid? Supercritical fluid is nothing but it is a fluid which shows the properties of both liquids and gas. So if we have some kind of fluid which shows the properties of both liquid and gas, we will say it supercritical fluid. And the process to make supercritical fluid is known as supercritical process. So this is a method in which we heat and pressurize the a material so that it becomes supercritical fluid. It is also used to break down organic materials such as biomass by subjecting them to high temperature and high pressure conditions. So if we provide high temperature and high pressure to it, it will also break down the bio. It will also break down the biomass into smaller and simpler molecules. And at last, gasification. Gasification means converting solid or liquid organics such as biomass into a gas mixture. So yes, it will go at the term. It will convert. It is used to convert any type of biomass into gas mixture, such as syngas, by exposing them to limited amount of oxygen in a high temperature environment. The syngas can then be used as a fuel or feedstock to producing chemicals. But if we look at the pulping is the breakdown of lignocellulosic materials such as wood or into constituent fiber, exposing them to chemical heat or pressure. So it may require chemical, it may require heat, it may require mechanical. It is not necessary that we need chemical and heat. We can also uh, use mechanical, and it does not break down the lignocellulosic material. Well, the things which we have seen, hydrothermal and supercritical gasification, these all break down the biomass into smaller. We have a big molecule and we break down to form it smaller. But pulping is not used for breaking down. Pulping is used to separate lignins. Lignins, we have a biomass and we will separate it into cellulose, hemicellulose, lignin, and extractives. So pulping is different from gasification and these things. In pulping, we do not break down into smaller compounds. We just separate the lignin from the hemicellulose, cellulose, and extractives. So that's why pulping is a very different kind of process when compared to gasification, hydrothermal treatment, and supercritical processes. And as we have discussed this also, that sucrose sugar is considered as, yes, it is considered as disaccharide, but the answer with the NPTEL was given as monosaccharide. So some students have said that they have selected the answer as disaccharide, but it showed mono. But do not think about it much. We will contact the NPTEL team and ask them to rectify the answer. So the reasoning for this is that sucrose sugar is a disaccharide composed of glucose and fructose unit. So it is a disaccharide and made up of glucose and fructose. So these two are monosaccharides and these are joined by glycosidic bonds. If we join these two, glucose and fructose, by glycosidic bonds, we get a disaccharide, and that disaccharide is known as sugar. So that's why it is not a monosaccharide; it is a disaccharide. So these are joined by glycosidic linkages between the anomeric carbo atoms of the two monosaccharides compound. So, thank you for today. We have completed our uh, week 3 assignments, and next week I will discuss about the week 4 assignments, and I will also try to convey about assignment 5 2020 year 2021 and 2022 in next week. And if you have any kind of doubts, you can always email me. I am sharing my email address in the chat. I have shared my email address in the chat, and you can always contact me if you have any type of questions.